diamond, then upload the diamond mechanized files. So to the Megan software, where you can visualize the results and analyze different results as per your goal. Now, the steps, this is just one of the summarized way of or graphical way of real life work which I have done it. So for another project, I'm not going to mention whose project was it, but those people who are aware and whose project was, he could easily identify that this method I have implemented for his entire projects. And simple way here, what are the database data sets you need it? For example, nr.tv, you will need it. This is the first file. Then you will need your own file, and then you will need to go for using the Velvet G or Meg Diamond and the Megan software. So, and then the different steps, what are the output which you are supposed to receive are mentioned here. For example, the taxonomy, seed, eggnog, interpro to go, like functional analysis and annotation things, you will be getting it done here. Comparison could be, for example, here there were two different types of samples from two different places. So we are saying inter-sites between infection stages and intra-site and between infection stages. So these are the way which I have used to compare and implement the method other than the most common method, which is KIME pipeline. So I have not used KIME pipeline, which is most common, and that is available on EBI website. But I have used a manual method, which was much beneficial and easy than the KIME databases. But the approach remains the same for KIME pipeline as well as when you are using Velvet G, Diamond, and Megan. Both the approaches have similar goals, and I personally believe that Velvet G is much better and easier to use it. Now, if you want to see it in graphical way with the command names in different steps, this is also one of the presentation. Then this is in the bottom of this slide. You could see how the output files could be visualized and analyzed. So this is like basically phylum, genus, species of all the microbial organisms could be analyzed, which I am going to show you in detail on the next slides also. So for example, if you want to go for comparative taxonomic analysis for each sample or for all the samples in one figure, you could go, for example, 3 samples were analyzed here together as a comparative study, or 6 samples were analyzed. Then you can see 1 color, 2, second, third, fourth, fifth, sixth. So all the different things, 3 colors, 6 colors, represents different samples. And the samples, for example, these are the colors which I have used to label it. And then you can see the difference in terms of graphical way of representation at phylum, genus, or species level. Then another way is to go for analysis of the total number of reads, taxonomy, seed, eggnog, interpro to go, and then what are the goals which you could achieve. For example, generally in metagenomics, the people are interested at phylum level, genus level, and species level. These are the 3 main parts where the people are interested. And then you can also make a kind of UPGMA tree for your samples, how it looks like. So this sediment means soil sediment. So you should be aware these are the samples names for different conditions. Now, the output could look like, this is another way of presenting the functional analysis of metagenomics data, and then you can plot them for different samples. This is sample 1, 2, 3, 4, and 5. So for different samples, you can plot the functional analysis in this way or in the other way as you want. Then phylogenetic diversity could look like at different level, for example, phylum, genus, species. So all the three levels of analysis are present here. And finally, you can summarize the tools in metagenomics, which are commonly used: KIND, Meta, PHL, AN, and MGRAST, Kraken, MegaHit, then Spades, Blast, Human, Diamond, Mega. And there are some more small softwares, which is quite easy and helpful to explore the analysis of all the metagenomics samples. Now, if you go for one line memory of the steps, what are the steps? That is sample collection, first step, then DNA extraction, then sequencing, then quality control of the sequenced file, and finally go for assembly, and then annotation, taxonomic analysis, functional analysis, finally the graphical visualization by using Migan, Corona, or R packages or other codes which you could write in your own language. Finally, the graphical representation of metagenomics study analysis pipeline could look like sample collection, DNA extraction, then assembly, sequencing, annotations, taxonomical analysis, functional analysis, visualization. And these are all overall summary introduction about metagenomics and the pipeline. And finally, if you want to see, this is the live example for the pipeline, how we run or execute the commands in metagenomics study. Then these are just one of the overview, and that's it. Finally, if we summarize it, it is a powerful, metagenomics is a powerful tool for understanding microbial communities and their roles in various ecosystems. Its application could be worth for health sector, agricultural purpose, environmental purpose, biotechnology application, and understanding and exploring the marine biology. Finally, the references could be many more, but I have just given 2, 3 references here. So you could also look over the previous slides and presentations where there are some more related topics, and that is always worth. Thank you very much. So the next part will be metatranscriptomics, and then de novo sequencing introduction. And finally, after that, we will be moving to